We are interested in the robustness of oriented tissue deformation during animal development. By oriented tissue deformation, we mean the extension of a tissue along one axis and a contraction along the perpendicular axis. Oriented tissue deformation is a key process during animal development. For instance, it's important for the head-to-tail body axis elongation and for organ formation. Oriented tissue deformation can be driven either by externally applied forces or by internally generated active stresses. In the case of internally generated stresses, their directionality needs to be controlled by some kind of orientational information throughout the tissue. Such orientational information can be provided, for instance, by cell polarity. Indeed, it is known from a number of biological systems that cell polarity controls active anisotropic stresses, which then drives anisotropic tissue flows. However, if you combine these ingredients into a computational model, we find that such a tissue would be unstable. This is due to a classical active matter instability that has been known in the field for over 20 years now, and it arises because tissue flows conversely affect orientation information. Now, this instability raises the question of how oriented tissue deformation can be robust during animal development. So how is this instability prevented during oriented tissue deformation? While there could be several possibilities, here we focus on the question whether morphogen gradients could stabilize the process. Indeed, we know from a number of biological systems that morphogen gradients can affect the orientation of cell polarity. However, whenever we have tissue flows, those will most likely also affect the morphogens, leading again to a feedback loop. We ask here under which conditions such a system can be robust. To address this question, we study the interactions between a scalar field describing the morphogen concentration, a polar field describing cell polarity, and a vector field describing tissue flows. Scalar field follows standard advective diffusive dynamics. Polar field also follows standard dynamics, but with an additional alignment term to the gradient of the scalar field. The polar field then defines an active anisotropic stress, where depending on the sign we have a gradient-extensile or gradient-contractile system. This active stress then drives viscous, incompressible flows. We then studied in detail the stability of this system, and we found that while gradient-contractile systems are generally unstable, gradient-extensile systems can be stable. When comparing these results to the biology literature, we find that there are many examples for gradient-extensile systems, however there are almost no examples for gradient-contractile systems. This suggests that the active matter instability that we study here acts as an evolutionary selection criterion. In other words, there's a developmental principle here that's directly rooted in active matter physics. We also studied the influence of other factors on the stability of the system. For instance, the polarity could follow a dynamics where it tries to attain a finite preferred magnitude, or where it tries to attain a zero preferred magnitude. In the case of a finite preferred magnitude, we found that the system is less stable than in the case of a zero preferred magnitude. Finally, we identified a few parameter regimes where the system has perturbation modes that grow only transiently. Such time-dependent growth rates of the perturbation modes are created by a distortion of those perturbation modes due to a deforming system. More details on these results can be found in our publication.